If you think about the animal kingdom, there is this, well, almost magical ability some creatures possess, regeneration.
Yeah, it's incredible.
It's the ultimate sci-fi capability, really. The power to regrow what was lost, perfectly restore damaged organs, or, effectively hit the biological reset button after a major injury.
It really is biological perfection. I mean, not just patching the hole, but replacing the architecture, like flawlessly.
Yeah.
For humans, it's always been relegated to the realm of fantasy because, well, we scar. That's our default.
Exactly. But today, we are diving deep into research that suggests the human potential for regeneration isn't necessarily gone. It might just be deactivated. We are unpacking a phenomenal study published in Science back in June 2025, work done by scientists at the National Institute of Biological Sciences in Beijing.
Right, I remember seeing that. Really groundbreaking stuff.
Their mission was pretty straightforward.
Yeah.
Figure out how to flip the regeneration switch back on in a mammal.
And they provided a remarkable proof of concept. I mean, this work is the first demonstration that one highly specific regulatory adjustment, one single genetic tweak, can restore significant complex regeneratability in mammals.
Okay, let's unpack this entire process then and trace the steps they took, because this isn't just about, patching up a simple cut.
No, definitely not. We have to start with the fundamental biological contrast, really.
Okay.
When you look at the true regeneration masters, salamanders, certain fish, or even some crustaceans, they can often repair structures like entire limbs or spinal cords without skipping a beat. It's just their default setting.
They truly hit undo. It's amazing.
Yeah.
And we know some mammals have very specific limited regeneration, right? Like deer regrowing antlers annually.
Yeah, which is an amazing feat in itself.
But they grow from a permanent bony base. The vast majority of mammals, including us, and importantly the standard lab mice used in research, we default to fibrosis.
Fibrosis, yeah, that's our problem. When we get injured, instead of rebuilding the specific tissue, the muscle, the skin, the cartilage, we rush to fill the void with this tough, dense, collagen-rich scar tissue.
It's a quick fix.
It's a quick fix, exactly, to maintain structural integrity.
Yeah.
But it often leaves the organ functionally compromised. Not ideal.
But the sources noted that scattered throughout the mammalian family tree, there are these outliers that do manage to heal well.
Right.
The team focused on these regenerative champions, species like the African spiny mice, rabbits, and even goats, which can repair tissues like ear punctures without forming those debilitating scars.
And this gave them the perfect setup for comparison. They called it the ear experiment.
Right.
They punched small, identical holes in the ears of both these regenerative animals and the non-regenerative controls, like standard lab mice and rats, and then just watched the healing process unfold.
And here's the key finding, at least from that initial phase. The process started exactly the same way in all of them.
Yeah, that was crucial. Both groups initially formed a blastema.
Okay, what exactly is a blastema?
So a blastema is the critical first step. Think of it as a temporary organizational hub. It's a cluster of specialized cells that kind of revert back to a more flexible stem cell-like state.
Like hitting a reset button locally.
Exactly. It's like the body hitting a temporary factory reset, getting ready to start construction fresh.
But that's where the scripts diverged dramatically, wasn't it?
Completely. In the regenerative species, that blastema got the go signal and carried on, meticulously rebuilding the complex structure, skin, hair follicles, cartilage, the works.
But in the non-regenerative lab mice?
Well, the reset button was pressed, but the construction crew basically never showed up. The blastema just sort of fizzled out, and the body reverted back to its default scarring mode.
So the potential was there, the reset happened, but the instruction manual stopped halfway through.
Precisely. The instructions were missing. So the next step was obvious. Find that missing instruction manual.
Which meant diving deep into the cellular conversation.
Yep. The researchers used some really complex tools, things like single-cell RNA sequencing and spatial transcriptomics.
Wow, those sound incredibly specialized. Why did they need that level of detail?
Well, they weren't just checking which genes were present. That's relatively easy. They needed to see which genes were actively talking or being expressed, and crucially, in which specific cells, like the fibroblasts, involved in healing.
Okay.
And at what precise moment after the injury. They had to map the entire communication network of the cells responsible for closing the wound.
Comparing the conversation in the mice that healed perfectly versus the ones that scarred.
Exactly. It's like reading the detailed script of every single cell right up until that point where things went differently.
That's amazing detail. And what did they find?
And that ultra-detailed view revealed one central regulator that was dramatically different between the healers and the scar formers.
Yeah.
A gene called ALDH1A2.
ALDH1A2. Okay, so if we zoom in on that specific gene, what's its actual function in this whole healing cascade?
Okay, so ALDH1A2, it encodes an enzyme dehydrogenase 1A2. Its job is basically conversion. It takes vitamin A
Right.
and converts it into a really potent signaling molecule, retinoic acid.
Ah, retinoic acid. And that's the huge deal here, right? It's way more than just a vitamin A derivative.
Oh, absolutely. It is the master blueprint regulator. Retinoic acid is one of those powerful developmental signals in all of biology.
Developmental, like in embryos.
Exactly. It's essential for embryogenesis. It guides cells, tells them how to organize themselves into complex tissues, where to grow, what identity to adopt, be it bone or nerve or skin.
So without that powerful specific signal,
Those temporary blastema cells, they just lack the architectural guidance they need to rebuild the correct structure. They basically lose direction.
wow. So the crucial difference wasn't a lack of building blocks, but a lack of management, a lack of instructions.
Precisely. The regenerative mammals, like the rabbits, they flipped ALDH1A2 on strongly right after the injury, flooding the wound site with that retinoic acid blueprint.
And the lab mice.
In the lab mice, that ALDH1A2 activation was, well, barely noticeable, almost silent.
And they proved this.
The proof was pretty undeniable. When they artificially supplied retinoic acid to the ordinary mice, their tissue repair improved significantly. They healed better.
Okay.
And conversely, if they used a drug to block retinoic acid signaling in the normally regenerative rabbits and spiny mice,
Their healing got worse.
Yep, their ability to heal was impaired, forcing them to default back to scarring, just like the lab mice. It really confirmed that ALDH1A2 was the critical gatekeeper, determining the entire outcome.
Okay, but wait a second. If ALDH1A2 is the key gene, why didn't they just look for, like, a deletion or mutation in the lab mice? Because the source material tells us something even more subtle and, frankly, more hopeful.
Yeah.
Both the regenerating animals and the scarring animals carried the same identical ALDH1A2 gene.
Yes. And this is where the research leaps from just interesting biology to a potential translational breakthrough. The gene was present in both groups.
Yeah.
The difference was all about how the gene was controlled. The key lies in the DNA enhancers.
Enhancers. Okay, can you explain those simply for us? What are they?
Sure. Think of the gene itself, ALDH1A2, as a powerhouse. The enhancer is like the master electrical switch for that powerhouse.
Yeah.
It determines when and how much power the gene should generate. So the rabbits and spiny mice had functional enhancers that were basically designed to flip the ALDH180 switch to maximum output immediately following an injury.
But in the lab mice, they had similar enhancers. The potential switches were there.
But they were just sitting there inactive, taped in the off position, essentially.
Precisely.
Precisely. And this suggests that maybe during evolution, perhaps to save metabolic energy or maybe to prioritize faster if imperfect wound closure mammals like mice and perhaps humans developed a mechanism that silenced this regeneration pathway.
So they didn't delete the file.
No, they just disabled a shortcut to open it.
That evolutionary implication is just astounding. That our own DNA might contain these sophisticated healing instructions we simply stopped using.
A powerful idea.
So tell us about the ultimate breakthrough experiment then. They took those functional enhancer switches from the rabbit and actually spliced them into the genome of the lab mouse.
They did. And the outcome was, well, pretty revolutionary. By installing the rabbit's active enhancer, the lab mice started producing high levels of retinoic acid following an ear injury, just like the rabbits did.
And the result on healing?
The result was significantly improved regeneration of the complex tissue structures in the ear. They saw new cartilage, new hair follicles, where previously they had only ever formed scars.
So they effectively awakened the dormant healing potential in a mammal that had seemingly lost it.
Exactly. They reactivated the pathway.
Incredible. So what does this all mean for us? This is the critical next step, right? Are we talking about regrowing a lost hand in the next 5 years?
Okay, let's manage expectations here. Not right away, and it's important to understand why. Unlike, say, a salamander, adult humans don't retain a large, readily available pool of highly flexible, multipotent cells. Our cells are already highly specialized. Skin cells are skin cells, muscle cells are muscle cells.
Makes sense.
So massive structural reconstruction, like a whole limb, is much, much harder. Simply activating ALDH1A2 in an adult limb won't instantly provide the necessary cellular raw material to build it.
Okay, so the path forward isn't just one magic switch then. It's more complicated.
Exactly. This research strongly suggests the true potential lies in a combined approach. Future therapies would likely involve, first, maybe providing a fresh pool of undifferentiated cells.
Like stem cell injections.
Perhaps, yeah,
Okay.
through stem cell injections. Then you would use genetic or epigenetic reprogramming, like reactivating this ALDH1A2 pathway we've been talking about.
Okay.
And finally, you need to provide the right molecular cues, like retinoic acid, to correctly guide those cells, telling them how to rebuild the missing structure properly.
So the immediate application then isn't the spectacular regrowth of limbs, maybe, but tackling those incredibly debilitating and common issues where fibrosis, that scarring, is the actual deadly enemy.
That's exactly where this work could be instantly high impact. Think about the possibilities for heart repair after a heart attack.
Right. Current treatment leaves scar tissue.
Which is non-functional. It weakens the heart over time.
Right.
If we can activate this pathway, maybe we could regenerate functional heart muscle instead.
Why, it'd be huge.
Huge. And the same logic applies to, say, slowing down kidney failure by replacing damaged nephrons, or preventing the devastating buildup of scar tissue in the lungs or the liver that ultimately leads to organ failure. This research potentially provides a fundamental map to overcoming fibrosis in pretty much any mammalian organ.
This is truly a remarkable achievement. So just to recap the core finding, we've learned that complex regeneration isn't necessarily lost forever in mammals. It might just have been deactivated over time. And crucially, scientists successfully switched it back on, at least partially, by manipulating a single critical control system, that ALDH1A2 gene and its dormant DNA enhancers.
Yeah, the profound implication here is that these complex, sophisticated healing instructions were actually maintained throughout millions of years of evolution, but their execution was somehow suppressed.
But science has now provided a mechanism.
A potential way to bypass that evolutionary decision.
Yeah.
And tap into this powerful built-in ability to heal complex damage much more perfectly.
Okay, so here is a final provocative thought for you, our listener, to mull over. Given that ALDH1A2 and retinoic acid are such extremely powerful developmental signals, we call them the cellular blueprints, what are the practical and, let's be honest, therapeutic challenges of controlling such a potent signal in an adult body?
That's the key question moving forward. How do you ensure that it only directs flawless repair where you want it and not, say, potentially dangerous uncontrolled growth elsewhere?
Yeah.
Getting that balance right will be absolutely critical.
A fascinating challenge for the future.